Hello students, welcome to EPG Patashala. I am Gaurinath from School of Life Sciences, Jawaharlal Nehru University. Today, we are going to learn on the module of protein structure in health and disease. This module is from biomolecules and their interactions. So in this module, we'll learn about how mutations can impair a protein structure and function with examples. The diseases result from the anomalies in this structure and conformations of proteins with examples. Protein structures plays a key role in the majority of human diseases, whether studying the target of an antibiotic or a defective protein resulting from a genetic mutations. Proteins are the basis of biology and target for most of the therapeutic strategies. When proteins become structurally abnormal, thus disturb the function of the cells, tissues, and organs of the body. The conformational diseases, many of these result from abnormalities in the structure and function of these proteins. These effects may be profound, such as structural changes observed in neurological diseases, sickle cell anemia, or may involve more subtle modifications of mutated proteins that are not correctly localized within the cell. Examples: Alzheimer's disease, Parkinson's disease, and Crawford-Jacob disease, cystic fibrosis, etc. So, as we are discussing about the diseases related to the mutations, here the example of sickle cell anemia. You can see the details in module 8, but briefly introduce here. In hemoglobin, where glutamic acid on beta chain, the sixth amino acid, which is mutated to valine. So this point mutation, which results to red blood cells being sickled, and that's why it's called sickle cell anemia. These sickle cells are deformed and rigid, which causes pain, inflammation, and which also damage the erythrocyte membranes, and due to which this hemoglobin cannot carry any more oxygen. Defective RBC is fragile and shorter in lifetime. A patient who has this disease is anemic, and it's a very lot of load on bone marrow, which produces the RBC. So what happens here is you can see the hemoglobin structure at the left side, where the green color is shown, that is the glutamic acid is became valine there. So when the T state, when the hemoglobin releases the oxygen and becomes a 10 state, where this valine of one chain and the similar kind of valine on the other chain, which come and interact, and they interact with each other, they form a fibrous structures. Now these fibrous structures become insoluble. They again don't dissolve back into the red blood cells, and due to which the RBCs become fragile, and the carrying capacity of oxygen goes, and those red blood cells not useful anymore. So this sickle cell hemoglobin is one of the responsible structural loss, due to which the functional loss. And as I said before, you can see more details about this in module 8. This one is disorders of collagen. Again, this one you can see the details of the collagen structure in module 8, module 6 of the fibrous protein. Tell you briefly here. So as the collagen is a building block for skin and several tissues, so the disorders of mutations leading to significant deterioration, the mechanical integrity of these tissues. So some of the diseases which are related to collagen dysfunction is Ehlers-Danlos syndrome, where muscle-skin junction disorders takes place. The other syndrome which I've listed here is osteogenesis imperfecta, where this is also called as a brittle bone diseases, where with certain kind of mutations in type 1 collagen, the bones become very fragile, and those people with these kind of mutations where the bones keep breaking and they also don't grow much longer, longer. They're generally short in height and they also have problem of hearing loss. Prion disease, it's one of the disease which is related to protein misfolding. Prion protein is the human gene encoding for the major prion protein, which is generally written as PRP. Expression of the protein is most predominant in the nervous system but occurs throughout the body, but it is more concentrated in the neuronal cells. The protein can exist in multiple isoforms. The normal one, PRPC, and the misfolded version is PRPSC, is associated with the conjunctive disorders and neurological diseases. PRP is highly conserved throughout the mammals. The precise function of PRP is still not very clear. So the prion protein, which we generally symbolizes as a PRP, which has a 253 amino acids, that's a coding region. But the signal sequences in the N and C terminal ends are removed post-translationally, resulting 208 amino acids. So there are several possible functions of the prion protein is described in the literature, but exact function is not known. So here I explain you one of the possible function seems to be the transport of ionic copper to the cells from the surrounding environment, or cell signaling, or in the formation of synapses. So this is a membrane anchored protein found mainly on neuron surfaces. So the proposed antioxidative function is this prion protein binds to copper and it has antioxidant activity which is resistant to oxidative stress and which prevent neuronal dysfunction. This is the most expected function of this protein. This is protein structure consists of globular domain. The prion protein structure consists of globular domain with 3 alpha helices and a small 2 antiparallel beta sheets. In the right side, you can see the 3-dimensional structure where the 3 alpha helices and 2 beta sheets and large loop regions are shown. The left side, it's a schematic picture where you can see the signal peptide, and the middle is expected to bind to copper ions. There is a hydrophobic part after the copper binding region, and the structure available, which is shown the right side, 3-dimensional structure, is the one between the copper binding site to 200 and about 240 residues. So this part, which is structure is determined, has 3 alpha helices and 2 beta sheets. So the one which we have seen, the crystal structure of prion protein, is the left side where it has a 4 alpha helices, and this is the soluble conformation of the protein where we have about 43% alpha helix, 3% beta sheet, and most of it is random coil or a coil region. The right side is the one which is a proposed model where insoluble fibrous protein structure it forms. Here, the alpha helical conformation goes down to 21% and the beta sheet conformation is increased to 54%. And these beta sheets are expected to stack with each other and form a longer fibrous polypeptide. Abnormal structure of PRPSC is responsible for the disease of prion proteins. The same sequence as PRPC are just different conformation, generally not seen post-translational modification. Deposited in cytosolic vessels, not anchored to the cell surface membrane as native protein. It is insoluble and forms large aggregates. Cells attempt to degrade PRPSC but can only remove N-terminal 67 residues, leaving the protease-resistant core with high beta sheet content, which is insoluble and which forms a larger filament structure when it accumulates. H protein, which is a soluble part. And these protein, when come across the infection, are that is a fibrous prion protein, then this globular part immediately becomes into the fibrous part. The fiber elongation takes place, and this induces the conformation in the globular protein to form a beta sheet rich protein, and it assembles onto the fiber. So these fibers, when they become longer, after certain length, they break, and they could act as a new seeds to accumulate or aggregate more prion proteins to form another filament. So these proteins, which actually the wrong conformation one is pushes the right conformation one to become a wrong conformation, and this one forms a longer filament. So this is a self-creation of, or conformation induced by the wrong guy influences the right guy. Prions aggregate with the within the central nervous system to form plaques known as amyloid, disturb the normal tissue structure, which cause neurodegenerative disease. This disruption is characterized by holes in the tissue, which results spongy structure in the neurons. Other changes include absence of an inflammatory reaction. Neurodegenerative symptoms can include convulsions, dementia, um, balance and coordination dysfunction, and behavior or personality changes. While the incubation period for a prion disease is relatively long, up to 5 to 20 years, once symptoms appear, the disease progresses very rapidly, leading to brain damage and death. Another protein causing diseases is Alzheimer's disease, and this is the most common cause of dementia, which describes the memory loss and difficulties with thinking and problem solving. These symptoms occur when the brain is damaged. Alzheimer's disease is one of the major reasons. Alzheimer's disease is named after the scientist Alois Alzheimer, who described it first, and this develops gradually, damaging more parts of the brain. Uh, it is a progressive disease, generally expected to happen at the older age, and this disease causes nerve cell death, tissue loss throughout the brain. Autopsy of a dead person's brain who is affected by Alzheimer's disease always found tiny inclusions in the nerve tissue called plaques and tangles. There are two different kinds of materials. These plaques are found to have um, between the dying cells in the brain, and which are made up of a beta amyloid protein. These are also called as amyloid plaques. Whereas the tangles are found within the brain neurons, and these are made up of fragments of other protein called tau. So these two are made up of different proteins, and their location is also different. The amyloid plaques are between the dying cells, and the tau tangles are found inside the neuronal cells. The beta amyloid is a fragment from a large protein, which is called amyloid precursor protein, in the fatty membrane surrounding the nerve cells. So apolipoproteins, especially APOE4, enhance the breakdown of amyloid precursor protein. So the amyloid precursor protein is cleaved by beta secretase, and the beta amyloid builds upon to plaques. APP is cleaved by beta secretase, and gamma secretase result in a beta amyloid polypeptide. This beta amyloid polypeptide is expected to form longer fibrils, which I will show you in the next slide. The small clumps of beta amyloid may block the cell-to-cell signal at the synapses. They also activate immune system cells that trigger inflammation, which engulf disabled cells. Beta amyloid peptide is very hydrophobic in nature and forms plaques with antiparallel beta sheets. The small peptide from 16 to 22 is enough to form amyloid fiber on its own. And if you see the right side, the structure of it, so if you have a the beta amyloid polypeptide, which is cleaved, so if you have from 10 to 40 amino acids, This is what which forms a small antiparallel beta sheet, and these antiparallel sheets stack with each other to form a larger fibrils. And this is the one which forms a insoluble plaque, which hinders the communication between neuronal cells. Tangles of tau protein. Tangles are made up of tau protein, destroy the vital cell transport system. The transport system is organized in orderly parallel strands, somewhat like railroad tracks. Cell organelles and other key materials travel along the tracks. Tau helps the tracks stay straight, and tau collapses into twisted strands called tangles, and the tracks can no longer stay healthy. They fall apart and disintegrate. Nutrients and other essential supplies can no longer move through the cells, which eventually die. So the microtubules function as the tracks for the synaptic vessels. So the microtubules function is regulated by bound tau protein, which you can see in the figure. The microtubules are stabilized by tau when it binds, and when the tau is removed from the microtubules disintegrate. So activation of microtubule associated protein or microtubule affinity regulating kinase, possibly by external insult to the cell, like the disruption of intracellular calcium homeostasis by amyloid peptides, triggers a phosphorylation of tau on its microtubule binding domain. So the phosphorylation triggers dissociation of tau from the microtubule surface, potentially destabilizing the microtubule. The dissociated tau further phosphorylated, first by cyclin-dependent kinase 5 and then glycogen synthase kinase 3. Once it is phosphorylated so far, this process becomes completely irreversible and it cannot go back again. The phosphatase cannot remove the phosphate and cannot send it back to go and bind to microtubules. Under this hyperphosphorylated tau delocalizes somatodendritic component and can subject to proteolytic cleavage. So once it gets cleaved, then it's no more functional. The resulting smaller and more aggregate prone fragments of tau begin to aggregate into oligomers, and which might further sequester soluble full-length tau and form the filaments, which are finally deposited as a neurofibrogenically tangles. So these small fragments of tau form like aggregates, and once these aggregates are formed, they act like a seed. Even they sequester the functional tau protein from the microtubules. Here I am again describing the amyloid fiber formation. The intracellular aggregates are a fiber tangles made up of protein called tau. The extracellular plaques made up of aggregates of beta amyloid polypeptides. That's we generally call it as a AB. So AB polypeptides generally have 40 to 42 amino acids, and the proteolytic cleavage of much larger polypeptide, which we talked about, amyloid precursor protein. These peptide is a very flexible structure and forms a small antiparallel beta sheet. In the right side, it is actually much better explained, where the beta sheets, 2 to the 40 to 40 amino acids, forms a small antiparallel beta sheet. They assemble together as a long fiber. They stack one over the other. They form a strong fiber. So it almost forms like a protofilament, and these protofilaments come together to form a large bundles. So anything that increases the concentration of AB peptides increases the possibility of formation of these protofibrils of misfolded protein, which will seed and initiate the aggregation cascade. So these sequester the actual functional protein and change their conformation. The large plaque seems to be responsible for neurotoxicity once these bundles form, and they are actually responsible for neurotoxicity. So once the seed forms, once the seed forms, and it actually accelerates the aggregation cascade. Parkinson disease. The early symptoms in this disease are movement related, like shaking, rigidity, slowness of movement, and difficulty with walking. And the later stages thinking and behavior problems, dementia commonly occur, and also depression. The other symptoms include sensory, sleep, and emotional problems. The shaking is the most common one. Pathology of the disease is characterized by the accumulation of proteins into Levy bodies in neurons and insufficient information and activity of dopamine in the midbrain. Diagnosis of Parkinson's disease is mainly based on the symptoms, neuroimaging like MRI being used for the confirmation. Generally, the anti-Parkinson medication is the dose of L-DOPA and dopamine uh, agnosis to improve the early symptoms of the disease. As the disease progresses and dopaminergic of neurons continue to be loose, these drugs eventually become ineffective. Parkinson's disease traditionally has been considered as a non-genetic disorder, but about 15% of individuals with PD Parkinson's disease have first degree relative who has this disease. At least 5% of the people are now known to have forms of disease but that occur because of mutations of one of the several specific genes. Like these genes code for alpha synuclein or Parkin leucine-rich repeat kinase 2, PTN-induced putative kinase 1, DJ1, and ATP13A2. We will discuss about a couple of them in this module. In general, people with mutations in these genes will develop Parkinson's disease. The most extensively studied PD-related disease genes are SCNA, LRRK2. Mutations in these genes, including SCNA, uh, SNCA, LRRK2, glucocerebrosidine, the GBA, have been found to be risk factors for sporadic Parkinson's disease. Mutations in GBA are known to cause Gaucher's disease. Parkinson is a component of a multi-protein E3 ubiquitin ligase complex, which is part of the ubiquitin proteasome system that directs the targeting of the proteins for degradation. So Parkin is a part of this complex, helps to degrade the proteins which are toxic to neurons. So mutation in the Parkin actually also causes this disease. Here show the structure of Parkin. The autosomal recessive juvenile Parkinson disease is due to the mutation in the Parkin protein. So according to one study, the patients with onset of Parkinson's disease prior to age of 40, and 10% of them are Parkinson's mutations in 18% of them, and about 5% of them are homozygous mutations. So the below 40 age, 10% of them are PD patients. Of the Parkinson diseases, have 18% of them have these mutations. If you see this one, the domain structure of the Parkin protein, and the last domain, the ring 2, where the mutation is generally seen, which causes the dysfunction. And when this doesn't do ubiquitination, and so the protein degradation is hampered, and those proteins start forms aggregation. So another protein which is responsible for Parkinson's disease is alpha synuclein. Is a protein that is abundant in human brain. It is found mainly at the tips of nerve cells in specialized structures called presynaptic terminals, and interacts with phospholipids and proteins. Presynaptic terminals release neurotransmitters from compartments known as presynaptic vessels. The release of neurotransmitters relays signals between neurons, and it is critical for normal brain function. Alpha-synuclein plays a role in maintaining the supply of synaptic vessels, help to regulate release of dopamine. Human alpha-synuclein protein is made up of 140 amino acids, also has few isoforms. The central hydrophobic part seen as in aggregates or fibril formation are Levy bodies, which are responsible for loss of neurological function and cell death. So again, this forms of fibrous structures which is responsible for loss of this neurological function. The events in alpha synuclein toxicity, the NMR structure is seen. You can see that it has two helices connected by flexible linker and large C-terminal flexible structure, coil structure. And so this protein is actually seen in non-beta compartment of Alzheimer's disease. Alzheimer's disease amyloid, originally found in amyloid enriched fraction. So this protein, what happens is that it slowly, once it's phosphorylated at serine 129, it starts forming a small beta sheet like oligomers, and it starts to form fiber structures. These fibers forms a Levy body, and this is the one which actually hampers the function of the neurons. So there are several ways one thinks that the aggregation of this alpha synuclein could be stopped by different kinase inhibitors and protease inhibitors, etc. So this is the Levy body which is responsible for the disease symptoms. The residues between 61 to 94, the central hydrophobic region involved in protein aggregation in the alpha synuclein. In the rare cases of Parkinson's disease, there are mutations in the gene coding for alpha synuclein. 5 point mutations have been reported so far. Alanine to threonine at 53rd position, alanine to proline at 30th position, glutamic acid to lysine at 46th position, histidine to glutamine at 50th position, and glycine to aspartic acid at 51 position. And genomic duplication and triplication of the gene appear to be a rare cause of Parkinson's disease in some lineages, although more common than the point mutations. These mutations in alpha synuclein may cause it to form amyloid-like fibrils, and that contribute to Parkinson's disease. CJD, it's again degenerative neurological disease that has no cure so far. It is again a fatal disease. They are also called as human form of mad cow disease. And CJD is caused by infectious agent called a prion protein. Prions are misfolded proteins that influence the properly folded counterparts to the same misfolded structure, which are similar to them. CJD causes the brain tissues to generate rapidly. The brain develops holes similar to sponge, and this could be responsible due to the consumption of contaminated food, sporadic in almost 85% of the cases. Some could be familial hereditary conditions. Initial symptom of CJD is a rapid progress of dementia, memory loss, personality changes, hallucinations, etc. The physical problems such as speech impairment, jerky movements, loss of balance, coordination, dysfunction, rigid posture, seizures also accompany the neurological problems. CJD was first shown to be transmitted by blood transfusions or tissue transfer. In familial form, a mutation in the gene for PRP or PRNP is responsible for CJD. It is also reported cannibalism has been implicated as a transmission mechanism for abnormal prions. The structural changes of prion proteins are discussed in the beginning of this module, where this prion protein, which becomes beta helix shaped structure and long fibrils. So this is basically responsible for this disease. Cystic fibrosis. This is another genetic disorder that not only affects the lungs but also pancreas, liver, kidneys, and intestine. Difficulty in breathing, coughing up mucus as a result of frequent lung infections. Other signs and symptoms include sinus infections, poor growth, fatty stool, clubbing of fingers and toes, uh, infertility in males, among other symptoms. Cystic fibrosis is inherited in an autosomal recessive manner. It's caused by presence of mutations in both copies of genes for the cystic fibrosis transmembrane conductance regulator protein. People with single working copy are carriers, otherwise mostly normal. They should not show any symptoms of the disease. CFTR is involved in production of sweat, digestive fluids, and mucus. If CFTR is not functional, secretions become thick. It is generally diagnosed by sweat, uh, sweat and genetic screening. Cystic fibrosis transmembrane conductance regulator. This is also called as a CFTR for the short form. This is a ABC transporter class of protein, um, which is an ion channel that pumps chloride ions and thiocyanate ions. Nearly 2,000 of cystic fibrosis causing mutations have been reported. The most common mutation is deletion of 508 position, results basically due to 3 restricted deletion in the gene, and loss of phenylalanine at this position, which causes the dysfunction of this protein. The mutant protein does not fold normally and it's very quickly degraded. CFTR protein is a channel protein. As I said before, it's a pump actually, which controls the flow of water, chloride ion, and thiocyanate the cells inside the lungs. So you can see the left side cartoon, the functional form and the non-functional form, where there's a small R domain because of these mutations, which actually blocks the pump and where it cannot pump out the ions which are not needed, and because of which you have disease symptoms. So students, let's summarize what we have learned in this module. Protein structure and function play important role in health and disease. Mutations in proteins not only responsible for the loss of function, they could also lead to irreversible aggregates, which could cause cell death. In the case of neuronal cell death or brain cell death, it will result in several neurological diseases. Such diseases we have discussed in this module. Many proteins, due to mutations, they lose the function and cause disease due to that particular function. The symptoms will vary according to mutations found in one gene or two genes. It also depends on a type of protein and its function. In some cases, due to mutations, degradation of proteins, they aggregate and form fibrils. The accumulation of fibrils cause loss of cellular function and result in cell death. And this way, the mutations not only cause a specific function of the protein, they could also lead to aggregates and cell death. Thank you.